Good morning as we start a new series learning all about DNA, RNA, and proteins. And in this section, we're going to start with an introduction. And in our introduction, we're going to go back and do a little review to see, do you remember what we learned about nucleic acid in the past? Remember, there's two types of nucleic acid. There's deoxyribonucleic acid, which is your DNA, which is what exists most of the time in your nucleus. And then there's ribonucleic acid, your RNA, which is made in your nucleolus. Both of these consist of nucleotides, which are sugar, phosphase, and nitrogenous bases. The sugar either being the ribose or the deoxyribose, and the nitrogenous, and the sugars are attached to phosphase, and those form that backbone. And then attached to that backbone are the nitrogenous bases. So let's look closer at those nitrogenous bases. So each of those two nucleic acids, DNA and RNA, will contain 4 of the 5 bases. So when we look at these 5 bases, you can see that 2 of them are double carbon nitrogen rings, guanine and adenine, whereas 3 of them, cytosine, thymine, and uracil, are single carbon nitrogen rings. Guanine and adenine can be found in both DNA and RNA, as well as cytosine is found in both of them. In contrast, thymine is found only in DNA and uracil is found only in RNA. So basically, just remember that thymine is only in DNA and uracil is only in RNA. Now, for DNA, in order to get that equal spacing, we have to have 3 rings going across, which means one of the double carbon ring bases, guanine and adenine, has to pair up with one of the single carbon ring bases. And it's not willy-nilly. They pair up with specific partners. And so what happens is that guanine always pairs up with cytosine, okay? So whether it's DNA or RNA, guanine will always pair up with cytosine. Now in DNA, adenine will pair up with thymine. But in RNA, because there is no thymine, adenine will pair up with uracil. Okay. So very important to get those distinctions. There is no thymine in RNA, and there is no uracil in DNA. When we look at this base pair bonding, these bonds are hydrogen bonds. So if recall when we learned about bonds, there were ionic bonds and covalent bonds. An example of a very weak covalent bond were these hydrogen bonds. Okay, so these are weaker than any other type of bond. So if you look at adenine with thymine as well as adenine with uracil, there are 2 very weak hydrogen bonds for both of these. In contrast, cytosine and guanine have 3 weak hydrogen bonds. So there's no difference in the number of bonds between the AT and the AU, so that the DNA and the RNA bonding numbers and quality are exactly the same. Going back and looking at nucleotides, it's real important that you understand the nucleotides and the parts of it. We have the phosphate group. This is negatively charged. And so what that means is it's going to be bonding to other things that are positively charged, any other proteins that are nearby. We have that pentose sugar, whether it is the ribose or the deoxyribose, and then we have those base rings with their carbons and their nitrogens, and they are going to have hydrogen bonds with the other base rings if you are in DNA. So this double helix structure that we find in DNA, we're going to have two strands of DNA side by side, but they run in opposite directions. If you look at this one, you notice if you take one strand, you will notice that there is a 5' written at one end, and if you follow it to the other end of that particular strand, the other end has a 3'. The 3' end is the tail end, so we don't add anything at that end. All the adding of nitrogenous bases is always done at the 5' end. And so you notice the 5' end, when there's a double helix, they are opposite each other. So all the adding is done before the two strands are twisted around each other to form the double helix. So when we look at this double helix structure, we have a sugar, a phosphate, a sugar. There's going to be another phosphate, sugar, a phosphate on each side, giving us that backbone with a 3' and a 5' in opposite directions. Then we have our nitrogenous bases with A's and T's bonding, because this is DNA, and C's and G's bonding with double hydrogen bonds between the adenines and thymines, and triple hydrogen bonds between the cytosines and the guanines. So that's kind of the double helix structure of DNA. Looking specifically at the chemical bonds, don't forget we're going to get ionic bonds, especially related to the phosphates. We have a lot of covalent bonding going over here, especially with the nitrogens and the carbons and the rings. And then we also have the hydrogen bonds. So we have all the chemical bonds we have talked about well represented in our DNA. And so we had this slide before, but you see, as opposed to when we reviewed this before, we have added a lot more detail. So don't just stick with the original detail we learned, but now you need to add on to it, which is why when I say to learn every single week so that we can add on to it, you really need to do that and not try to wait until the weekend before a test and cram it, because we build on every single previous lecture. So now that we understand DNA and RNA a little better, let's talk about how we make DNA, RNA, and protein. To make DNA, we take pre-existing DNA and we're going to do a process known as replication to make new DNA. And then from DNA, we're going to do a process known as transcription to make RNA. And then from RNA, we're going to do a process known as translation to make protein. So let me talk a moment just about RNA. There are actually 11 different types of RNA in a cell. Now, we talked about how RNA is made in the nucleolus, and I want to talk about 3 different types of RNA, because we need 3 different types of RNA for these processes of transcription and translation, which we are going to be talking about in this section of the course. The first type of RNA we're going to be talking about is messenger RNA, and for that we write it with a little m and then a capital RNA. And messenger RNA is the type of RNA that's going to specifically be coding for the proteins. It is the type of RNA that we are going to be looking up on a chart. Okay, it is made in the nucleus and then has to exit the nucleus through one of those nuclear pores, and it's going to work when it's attached to a ribosome. And as you know, the ribosomes are going to be either free-floating in the cytoplasm or they're going to be attached to the rough endoplasmic reticulum. So the messenger RNA has to exit the nucleus and attach to a ribosome to work. The second type of RNA is known as ribosomal RNA, and this is the RNA that actually forms the ribosomes. And so the ribosomes are made in the nucleolus, and we have these 2 subunits. And if they were to join together in the nucleus, they would be too big to exit the nucleus. So the two subunits actually exit through the nuclear pore. And once they're in the cytosol, then they will join together in the cytoplasm, become one of the organelles. And then the messenger RNA can attach to it. Right along here, where you can see the two subunits. In this case, they've made the two subunits two different colors, so you can clearly see it's two different subunits. And the third type of RNA is called transfer RNA, because basically it's transferring other things to this complex of ribosomal RNA with messenger RNA. Okay. So when messenger RNA is attached to a ribosome, then transfer RNA is going to be bringing an amino acid, which is this little green ball at this one. The transfer RNA is this cloverleaf structure right here. And it is going to bind to both of these structures, and its job is going to be translating the message of nucleic acid into that, from that language into the language of a protein, the amino acid language. So it's got a real important job. So I talked about we're going to have translation, and I talked about transcription. So what basically are the differences between transcription and translation? And not to worry, each of these are multi-step processes, and we're going to go through them in great detail. Even though each of them have three very similar steps, they are tremendously different. So transcription is changing one type of nucleic acid to a different type. In other words, we're going to go from DNA to RNA. So we're going to take one strand of DNA, because remember, it's two strands. And we're going to take one strand and we're going to use it as what we call a template. We're going to take its nucleotides and we're going to make a complementary strand of RNA. So where there would be a cytosine in the DNA, we're going to have a guanine in the RNA, etc. And then that messenger RNA is going to leave through the nuclear pore so it can go in the cytoplasm. And then we're going to move to translation, where we can take the nucleic acid language of the messenger RNA and change it to a protein language in the cytoplasm. And that's going to happen by taking this and attaching it to a ribosome. And here's our messenger RNA attached to a ribosome. And here are incoming transfer RNAs, and each one is bringing in a little amino acid to form a new protein chain. So transcription occurs in the nucleus, and then the messenger RNA exits, and translation occurs outside of the nucleus in the cytoplasm and will involve the ribosomes. All right, that's it for this first section. I will see you shortly as we continue learning about DNA, RNA, and proteins. Thank you.